I think that's it. So with that, I'm pleased to present our speaker. We have Anthony Huffman today, who is a grad student in DCMB.
Okay. All right, just want to make sure that everyone in the room can hear me just fine. So I'm Anthony Huffman. I'm currently a second year PhD student in Oliver's lab. Most of my work for the past few years have been focused on either vaccine gene expression or helping out with ontology design related to CIDO, which I'm going to be presenting here today. This talk is an expansion of the one given during the International Conference of Biomedical Ontologies 3 weeks ago, and I'll be going in more of the histories in CIDO, in addition to some of the new updates that we've made. Okay. So CIDO was prompted by the COVID-19 pandemic that emerged 3 years ago. So I'm sure everyone is well familiar with the impact it's had. We increasingly moved into more of an epidemic phase from the pandemic, but it's still a widespread concern throughout, with still millions of new cases and few deaths emerging within the last few months. However, COVID-19 historically was not the first major coronavirus disease that we are worried about. We had the initial SARS epidemic that emerged in November 2002 in China, which lasted thankfully only 8 months due to very strong efforts made to crushing the disease, but it still had a significantly high case fatality rate of 9.6%. There was a second coronavirus infectious disease, MERS, which emerged back in 2012. Well, that one spread to a bunch of other countries and had a very high case fatality rate. It thankfully was able to be quarantined and reduced. And while these were the major coronavirus diseases that we need to work, is this moving automatically? Okay. There's still a variety of other coronavirus species which cause common colds and other less severe disease states in humans. So it's still a group of viruses that we need to be somewhat concerned about. So, again, I reiterated what's COVID-19, which really pushed this as being a priority to develop. So as part of this effort, our lab initiated the creation of the Coronavirus Infectious Disease Ontology. So we do so as part of the Open Biological Biomedical Ontology Foundry program, which is a collection of a large amount of ontologies focusing many different fields. And our intended goal for this project is that we integrate basically all relevant information relating to coronaviruses. So that relates to the actual etiology of the species, the different phenotypes we see both in the host and the actual disease progression, representation in different reservoirs, in addition to transmission across species, all of which are forms of host coronavirus interactions. We also are interested in data relating to the diagnosis of COVID-19, along with drugs and vaccines that are effective or that are designed to deal with actual vaccine status. Then third one that we were also looking at is the idea of metadata and data standardization, which is one of the main reasons we actually want to deal with this as an ontology. So for those who are unfamiliar, the modern ontology framework emerged back during the 1990s. The current definition of that is that if an ontology is any artifact, or that is a schema that has a set of representational primitives which are used to model a specific domain of knowledge or discourse. So what that means in simple terms is that ontologies tend to be directed knowledge graphs, which we link to various different nodes, which represent concepts to a common relationship. So one of the more common examples that we have of ontology is the idea of ISA and project relationship, where we assert that the ISA relationship represents the primary mode of inheritance. So child A is property of B, or let's say A represents cats and B represents pets. So we would, well, mammals more largely. So when we say that A is a cat, and then B is also a cat, and we say that all mammals are part of the biosphere, then we can also assume that any of the children properties will also inherit this property. So the reason why we are interested in this modern ontology framework is that it provides a very nice, logical, and consistent way to integrate and harmonize a wide variety of different data. This is especially more important with the emergence of big data, which is often really fragmented into different experimental annotations, different measurements, or even some experimental conditions. So if we're dealing with like a massive amount of studies or papers, and with COVID-19 having, I believe, over 12,000 different papers being published just now. Yeah, and I think that's like an old number.
I'm sure it's exponential.
Yeah, but so we have all these different papers on the same focus on the same idea, but they're using different methodologies, different focuses. So when we actually want to make sense of all of it, ontology provides a very useful tool in providing a consistent set of underlying axioms for both individual classes or properties of data. So this would be viral variants, viral species, but also if we look at instance level data, such as a specific sequence that we would get from a data set. The second use of ontology is that this logical framework also makes it much easier to integrate computers and machine learning when it comes to reasoning through these connections or making results. So ontology rests on an open world assumption, which essentially states that instead of a closed world, which is something is true or not true if you query it, ontology is capable of accepting a third category where unless otherwise, where unspecified is not the same as not true. Makes it a bit easier for inferences, especially if you're trying to look for patterns. The other one is that, as I mentioned before, any of the children class will inherit properties from part of across descendants. So if we annotate that COVID-19 S protein targets mammalian cells, we know that's going to be true for human cells, for dog cells, for deer cells, which applies to a wide variety of different potential reservoirs and other behaviors. And thing is, ontology use tends to be kind of hidden, or at least not really assumed, because most of our results either rely on ontologies as like basic framework, and we only want to plug in data. But we still have like three major examples of ontologies that I'm sure almost everyone has used or will have used for the last one. The earliest framework for classifying different organisms and stored by the NCBI taxonomy can be thought as one of the earliest bioethical ontologies. We have gene ontology, which is a standard used for any sort of functional analysis and trying to make sense of either gene expression pathways or other frameworks. And the third one, which is a basic formal ontology, is one of the more emerging ones, but have been recently accepted as an ISO standard for representing philosophical concepts, being on par of the actual meter or kilogram when it comes to standardizing metadata. So going on that, I'm going into one of the initial ontology design patterns that we started with CIDO. So this is back in 2020. It's gotten much more complex, and we're having more interlinking relationships. But the basic idea is that using SARS-CoV-2 as an example, is that we know that SARS-CoV-2 is a virus. We know that it infects some animal, which we can link to humans. Each of these humans has specific qualities that are either affected by a disease process, which are in turn regulated by genes. And of course, each animal as part of the disease process is being realized for a specific disease phenotype or it occurs in specific organs. And this disease process can be targeted by specific COVID-19 drugs and COVID-19 vaccines. So all of these relationships we can use to plug in and start to make sense of the actual metadata or further studies we want to go through. So the most recent, yes.
So I'm curious, like, I mean, this is really cool, right? I always think ontologies are really powerful foundation for a lot of stuff. I'm kind of curious though that there wasn't, or maybe there was and it wasn't good enough or whatnot, that viral infection ontologies feels like that would be a general, like there would be general ontologies for that already exist that you could then layer on more COVID related terms into that to, add some additional specificity to it. As opposed to like, virus versus like, or is that what this is? This plugs into a larger ontology.
I believe the timeline was that CIDO was published first, but we were working with John Beverly for the viral infectious disease ontology and the general infectious disease ontology.
Okay,
So in this case, I think we kind of were first to publish, but we are working with the infectious disease ontology and still ongoing.
okay, so but it's okay, that makes sense.
And we
Yeah.
had an earlier vaccine ontology.
Mhm.
Years ago.
Okay, yeah, I was just curious, because it feels like there, ontologies, there's lots of them. I just wasn't sure sort of where this, this was adding on to like, building upon earlier work to then. Okay,
So I'm actually going to get into that in the next slide where
Yeah.
I go into the.
I'm really guilty of that, asking questions that are literally answered on the left side, so I apologize.
It's good, Ryan.
Yes, that means that you preempt my presentation. So I believe this is a most recent version of CIDO, unless someone updated when I wasn't working today. And using the Ontology query, we have over 11,000 terms in total, representing a combination of different classes, object properties, and different instances, data types. So each class represents the basic conceptual idea that we're focusing on. So this could be a person, this could be a room, this could be a university. Object properties and if classes are specific nodes, object properties are the edges which connects nodes, which relate them towards each other. And while we have a much smaller amount of specific object properties, we can add them as specific axioms for each of the results. We have some further annotations where instead of object properties, which tend to be class level mappings, we have data type properties. Oh, okay, it keeps on moving with that. So, which tends to be focused more on instance level data, which again mentioned is specific details. But while we have this very big table, which I think is kind of hard to see, which gives the origin of each of the original ontology terms from the parent ontology. CIDO specifically has about 1,500 out of around 11K total terms. Here you can see major contributors over with Kebi, CIDO, NCP, taxon, a few others. I'll go further in as analysis, but. So when we take a look at the high level structure of CIDO, we see we have an example of various different classes and terms that are related. So each of these little terms in parentheses represents specific ontology and the origin of ontology. So DOID tends to be the one focuses human disease, infectious diseases, which I believe you were asking about through that.
This makes a lot more sense to me now. Gotcha. I see where this fits in the overall structure, so great, this is awesome.
Yeah, so the highest level ontology and part of the universal term we're doing is basic formal ontology, which is the main high-level framework that we use to try to keep all the different ontologies interoperable. Otherwise, you get into some mapping issues depending on specific terms. So while I won't go too much in depth, I'm just going to spread out the difference between continuing through these high-level terms. So entity is the basic high-level concept where we're dealing with any things, and it's split up into continuance, which are entities without a temporal component, identities with a temporal component for occurrence. So this could either represent a specific temporal region where we're talking about a day or let's say last week, well, or processes, which are events which have a bunch of participants that occur for some designated time period. Over here, we have material entity, we have qualities and realizable entities. Material entities are entities made out of matter, so this would be most of the animals, most of the objects, and the other details are popping in. Qualities are more of the adjectives or inherent traits, so being tall, being pale, or being hypertension, or specific other details from there. So these are descriptors mostly. Realizable entity is a bit more complex one, but these are latent traits which only occur due to interactions or results. So these would be your jobs, your professions, and it keeps on moving without my permission. So when we have all these different hierarchy and terms, and mostly want to show that CIDO is basically adding on to each of these frameworks in smaller details, focusing on greater specificity, but when it interacts with COVID-19 or other coronavirus diseases. So I'll be going more into these as part of the later ones, but just want to show the general framework. So going on to probably the most fundamental or most frequently interacted class in CIDO is when we actually deal with the specific coronaviruses. So what we mostly focused on for most of these results are coronaviruses that were either that had some vaccine component, since most of these terms were integrated due to it was framework of vaccine ontology. So while we have a couple of these for the common cold, which people didn't really care about, but what's going on, we can also see some of the MERS, SARS, and SARS-CoV-2 all emerging as specific diseases that we know to interact with humans and countries underlying pandemics. Again, we import these terms from another ontology, and as part of this framework, we primarily focus on importing only terms that are needed for the representation of concepts, and that if there's any underlying framework or other details or axioms assigned to the classes, we continue with them. So the second side is where we're focusing the imports when it comes to coronavirus phenotypes. So most of these phenotypes are relating to what we consider to be the disease process or disease symptoms. So we have different parts of fever, the cough, the headaches, and these are specific symptoms. Most of these are linked into either the acute COVID-19 disease or through the long COVID-19, where we tend to have more information on the acute COVID-19 due to it being easier to study. So moving on to the actual coronavirus genome, or in this case, SARS-CoV-2, since that's the primary focus of what we're mostly interested in these days. So as you can see, SARS-CoV-2 has a relatively small viral genome, about 10 to 11 so genes, and then a significantly larger amount of proteins which are set up. So normally ontologically, we try to set it up where we say that each protein is a product of one specific gene, and that if there's specific deletions or modifications of the protein product, then it's listed as subclasses. Now, due to part of the confusion or concerns with our genomes being very compact, is that we can occasionally have protein products which are frameshifts and contained within other inner genes, which makes it a bit more complicated since we're having different definitions of the gene as a specific informational content entity or genes as a specific set of nucleotides, which end up being used to produce a protein. So for CIDO, we focus on the definition of genes as just being a material entity of nucleotides, which correspond to a specific protein as part of OTG. So all the red boxes are proteins that could be produced through, some of them are either just putative proteins instead of actually known through. We have some case of like ORF9C, which might actually be have some specific effects when it comes to viral transmission. The second one that I want to bring up is some polyprotein replicase 1AB, or the rep gene translational project of SARS-CoV-2. So this is a frameshift polyprotein, in that we have a specific protein that codes for one large protein sequence. But around part of the time you get this frameshift, which ends up producing a second protein, which adds some debate on how to represent this ontologically. Protein ontology decided that the simplest way is just to say that any of the translational product actually pops up. It's just going to be the product linked to the specific gene. However, the polyprotein 1AB is actually an autocatalyzing protein, where you have the initial section removing NSP1, NSP2, and NSP3, then NSP3 cleaving the rest of the product to produce all the non-structural proteins which are relevant for viral replication and viral assembly. So I believe, which you can see, while most of the ones that are shared 6, 7, 8, and 9 don't have specific notations, the ones in the back half and then part of the frameshift have really important roles as functioning as a viral helicase as a proofreading exonuclease along with all the parts needed for biotranscription. Technically speaking, non-structural protein 11 would be considered only part of polyprotein 1A, but it's generally non-functional since the part that overlaps is NSP12, which is much more important. So going with all these proteins and genes, we then had a question on how to handle the different viral variants, which are popping up.
So, can I
Yes.
ask another question. So this is a lot of information.
Mhm.
This is like, even what you just said, it's a very complex system, viruses, just with overlapping programmed or non-programmed frame system.
Mhm.
When building an ontology like this, like, do you just have to manually curate papers that describe this? Like, where does this information come from?
So that is a good question. So part of it is manual curation during the initial steps where we decide the actual framework, how we're going to get it integrated and how we go for the results. I think almost all BFO ontologies have like committee members or organizations that manage to through updates, either through community talks through GitHub saying that, hey, this is wrong, or I need this term, and then we can add it or not, or through internal ones just going through what's part of the literature or primary results and for the actual annotation. So that gets into the second one, which is what do we use as sufficient evidence of proof or part of linking?
Yeah, that was what I was gonna say, like, ORF1AB encodes polyprotein A1A and 1AB and autocleaving, like, that information, like, is that proven? Is that one paper that said that? Is it 10 papers? Like, is it established? Like, how do you make decisions like that? I guess the committee or you
So,
just include everything and then weight it differently?
no, one part is part of the committee for how we're going to integrate or depend on those results. So protein ontology has a default pipeline when it comes to specific proteins, where if there's, hey, these are possible proteins, these might be putative, they could be added in, but then you would need actual experimental evidence and other primary sources before, okay, it's this protein, these are the annotations, and then that's.
So you set policy about what you decide to include as connections, and then, does this mean that is this like a living ontology where like some new paper comes out with some new link and some evidence, and then you go back and you make, this makes this or something, like some new connection in the ontology?
Yeah, I'm going to get to that at the end of that, but definitely one of the challenges with CIDO is that most of the ontologies, if you do a good job, are relatively well established, and you would only need to add some minor details. So basically, specific vaccines as they emerge, or and don't actually have to change the higher level structure. This one is because there's new information and we're getting new results. And this is probably where we go into the viral variants, because all the initial research and results were on the Wuhan strain.
Yeah.
So part of the living ontology that we had to handle is how to handle viral variants and how do we represent them, especially since viral variants are, I'm going to get into the next slide, a bit more difficult to describe ontologically. Since one of the principles of an ontology is that if you have an axiom, it must be true. So, which gets a bit difficult when we deal with susceptibility or other conditional factors, which do not always follow principle of being easily true.
It's really, it's a huge challenge, for sure. I was just reading last night that there's real concern about a bunch of new emerging variants this winter.
Yeah.
Most of which are, kind of on they're all Omicron related strains, but, showing more, ability to evade the immune system.
Yeah.
Some are BA2 related, some are BA5 related, and so on and so forth. So, and we know how this is all gone, that some of those earlier variants are really kind of in the history book,
Mhm.
I mean, it's really, it's emerging very quickly,
Yeah.
continuing to do so.
Yeah.
So I guess, is there lessons from flu or from, common cold, adenovirus or other viruses that are already kind of on this change? I mean, what the article in Science pointed out was that, coronavirus, SARS-CoV-2 and the Omicron strains
Mhm.
are acting like influenza and the common cold now. They're out there fighting their way into, survival dominance. So I just wonder if there might be some lessons there from some of the other work on ontologies and other viruses.
Yeah. So I would have to double check with CIDO what it's doing or IDO. Since again, most of the initial representation of the viral variants, CIDO kept on individually preempted a bunch of things that CIDO and IDO is working on right now. So through one of the other challenges that you bring up is with the burden of we're handling both of these results, which we can either handle specific annotation as instance level results if we want to focus on specific sequences. But the other big problem is that this is on the side of things that we've been relatively conservative when it comes to adding new information into the ontology when it comes to experimental, which means that when we have this rapidly newly emerging variants or some of the effects, or focus on the viral variant level, we are much slower when it comes to actually adding useful axioms because we need to actually test them. We need to make sure that we know it's specific to a viral variant. So we did come up with an idea to how to handle that, but I'm going to get to that later on. Since that choice is more interest is definitely one of the bigger challenges that we found. So when we're dealing with the broad idea of a viral variant for what's going on, we list them as all being a child of SARS-CoV-2. And that each of the specific viral variants has an amino acid variant, which is a specific mutation that is either frequent enough as part of the viral variant to be associated with. And when it comes to experiments or other analysis, we do have a subclass for a greater degree of correlation with each other, but we'll get into that later. So each of the specific amino acid variants, which we can end up annotating for specific properties, are in turn linked to their own protein. And so far, we've kept 3 major clades of viral variants because there's actually a few different ways to refer to them, which was another problem we had. So early on, and we have the GSI-8 clade, which was the first definition that I saw used in order to describe the viral variants, and was the most ontologically simple definition. Simply because it gave the definition of this is a clade member if it has this specific mutation. So like clade G only having a specific mutation for G614G and S protein, which made it relatively easy. And we were initially hoping that would make it a very nice framework. But unfortunately, this is mostly kept for legacy since GSI continued this back in last year as part of the naming schema. Instead, it's picked up the pango lineages, which are the BA2 ones that Dr. AP has already mentioned. Now, these ones are a bit more complex to define ontologically, because these are dynamic clades or lineages and ontologies want everything to be nice and consistent. So if you have a definition that's constantly changing, that causes problems for the reasoner and makes it much more confusing. Especially if we assert that Omicron or BA2 has these specific mutations in all their strains, which isn't true. Because when you look at Pango, they said, here are the mutations that we find in 75% of the sequences. And thing is, Pango uses a machine learning clustering algorithm in order to assign the actual properties. So while technically the definition is that this is a cluster that has been assigned altogether for the definition of Pango lineage, that's not like a really useful definition when it comes to doing any research or analysis. So, and of course, you also get the case of if we want to assert definitions of specific Omicron Pango lineages, you run into the issue of inheritance. So normally we say we have strain BA2 and then let's say strain BA2.25. So normally we would define if we say that BA2.25 is a child or is a strain BA.2, well, technically true if you take a look at like the the family tree of viruses, as it were, you run into the issue where he says, oh, BA2 has these specific mutations always. And then thing is, the new substrain variation is that it works by having a different amino acid variation to distinguish it from its parent, which means that we can't really infer that relationship. So which required us to create a new axiom for derives from as part of the to show this evolutionary relationship. Thing is, this is a placeholder term since BFO is currently having a debate over what's a proper relationship to define evolutionary constraints in this sequence. So we're keeping it until they finally decide. So the third category, and I think this is a version everyone is most familiar with, and these are the World Health Organization's classification schema. So these are the alpha, beta, delta, and omicron. And this was a problem earlier on because their definitions were, was that if it's part of this geoside strain or this Pango lineage or even next strain group, before they were all harmonized, you have like a really messy cluster of specific results. So that runs into the issue where we have specific descendants of the Pango lineages being folded up in addition to geoside clades, along with focusing on only naming once we get a specific variant of interest or variant of concern emerging. We also can't annotate that because that's a temporary thing. Alpha, beta, and gamma were all variants of concern, which no longer are because they've mostly gone extinct, or well, extinct in humans. There's still a possibility that they exist in some animal reservoir population, but for human epidemiology, they're no longer variants of concern. So our solution to handle this problem was instead of focusing on specific viral variants or annotating based off of viral variants, we would focus primarily on amino acid variants. So that is, we can link each of these amino acid variants, for example, D614G, and S protein to SARS-CoV-2 BA.2, and using the amino acid variant and relationship. So focusing all of these ideas, we can annotate each of these proteins with specific properties relating to its chemical composition. We could also annotate based off the experiments done on the individual amino acid protein to for studies relating to vaccine efficacy, to viral transmission or host pathogen interaction. So we can annotate all these details for the amino acid. And then if someone wants to do a query, they can say, okay, we have the sequence has this amino acid variant, and then we can infer properties from there. In this framework where we just focus on amino acid variants and then match it up to specific sequences, seem to be a much more sustainable method of handling the emergence of new viral variants. Now, so I'm sure you also noticed that we have all these material entities here, and then we have a bunch of red entities here, which seem to be here.
So the question,
Yes.
I'm asking questions because I'm interested, so I'm going to keep interrupting you. But this makes sense for condensing down sort of and consolidating. But I'm a nucleotide guy, so I have to ask, like, especially for things like, like you mentioned frame shifts, right? So there might be specific. I mean, I guess if something causes a frame shift, it's likely going to change the amino acid. But I was also thinking in terms of there may be things at the DNA level in the virus that RNA level of the virus that don't necessarily change an amino acid, but are relevant to the function of the virus. And so would that mean that that wouldn't be included, or just wouldn't be included in this sort of part of the class of the ontology?
So right now it wouldn't be included, since as part of just handling the actual experimental results, it was just much easier for us to use amino acid experiments instead of experiments testing specific nucleotides.
And it's likely more relevant too. I'm not saying, I'm just curious. I'm thinking edge cases here.
Yeah, no, that was something we discussed, and that actually related to the mutation side of the story. And this is actually something that we might end up changing in the next few weeks, since it was a major point of discussion for Denver Pre-DOS conference.
Okay.
So we have the fundamental material entity where we have the amino acid variant be defined as specific residues within the SARS-CoV-2 proteome. But then we have the underlying mutation event, which ends up causing a change in the proteome. And while this seems like a bit more of an academic point, this actually came up when we're dealing with deletions or nonsense mutations. Since for a changed amino acid residue, you need to have the amino acid residue still existing. So if you remove the deletion, it's no longer there. It's nothing.
I mean,
And-
technically it changed it to nothing, but yeah.
Yeah, but in which case, and then when you get into ontology and nothing, that breaks things because nothing can be reasoned to having any specific axiom or property.
And that goes back to the beginning slides that you
Yeah.
had where you talked about the difference between knowing something and inferring something
Mhm.
and not knowing something. So yeah.
Yeah, which means we had to basically create a parallel hierarchy of mutations into the actual SARS-CoV-2. Again, we focused on the idea of amino acid deletion initially as it was in parallel to the amino acid variants as part of our representation. But then we're getting into further discussions with either protein ontology's definition of deletions, which is just a truncated amino acid of everything around it. And we also get, as you mentioned, potentials in non-amino acid mutations within the DNA, which would end up affecting translation or transcription. So far, I haven't found any specific studies to add it to CIDO, but we're thinking about revamping and using this framework to, in case that does occur. So beyond that, the only other detail I want to mention is the use of characteristic amino acid variant instead of amino acid variant. So characteristic amino acid variant is a subclass of amino acid variant, which we say that the specific variant is used to identify a specific sequence as belonging to a specific viral variant. So that seemed based on how Pango lineages and other sets use the best way to handle the cases of this is how we know if it belongs to Delta or if it belongs to Omicron, which is saying that if it has these characteristic amino acid variants, then they can infer there. So again, there's still some potential issues if you get overlaps and only have like small amount of amino acid variant sequences, especially due to open mode ontology, but this is still the best framework we have. And of course, since all of the experimental annotation will be done on the amino acid variant level, it shouldn't cause any major issues. But if there is, then that's going to be another set of talks. So moving on to the epidemiology side of SARS-CoV-2. So we have the idea of specific lab samples and experimentations from either FDA or other government organizations. And then just specifications when we're loading in machines or doing analysis for blood plasma and further results. When it came to epidemiology, that came into another more complex issue, since epidemiology deals with waves or a pathogen occurring and then disapproving. So, and also raising the question of how do we axiomatically represent differences in specific reference strains and infectivity, since we have all these different factors to work out. So our solution for this was to just use the average RO value for a data property and simply assign each of the main strains the relevant RO so that the computers can reason out the actual infectivity rates. Again, we use average because we want it to be an axiomatic statement, and of course, actual RO can vary depending on public health interventions, vaccination rates, and population. So again, that's one of the biggest tensions we have, which is we have all these factors, but we want to boil it down to something consistent. Okay.
Oh my gosh, you know, it's just mind boggling.
It just keeps going deeper and deeper. And
Oh,
deeper.
you know,
Mhm.
it's nuts, really.
Yeah,
This is, I mean, recognize this is a significant amount of work, right? Like this is probably a lot of arguments and debates and fighting and nasty emails, but
Well, then getting people to really use it.
yeah. No, it's been fairly polite so far. We haven't had any fights at the last conference, so
makes them memorable.
Yeah, right.
there was one heated argument with definition of virus, but that's, I'll answer that later. So,
Anthony,
yeah.
there's a question online real quick.
Yes.
Patricia was asking how you define the edges, what's not included.
So are you talking about the object properties or that are connecting the different nodes or classes? Or are you talking about the edges of the definition for a viral variant? So,
Try to get her up on the screen, Marcy.
yeah.
Yeah, Patricia, if you have a microphone, want to unmute yourself, you're welcome.
Mhm.
What I was thinking is this is very similar to some of the work that we do in the library where we're not, when we are looking at, you've got all of these pieces and you have to decide the value of each concept that's added into the group, whether it's going to be a useful value to include. So I'm looking at all of these, and you talked about the messiness of long COVID, for example, and how ill-defined it is. And I'm thinking, where do you draw the boundaries on what you decide is useful to include and what not to? How do you define the edges of any part of this? I'm assuming the relationships are probably a little easier than the messiness of the entire universe of all of the terms and concepts.
So that's a great question. So there's two aspects to what's going on. One I mentioned is that if there's specific new concepts or edginess, typically we have members of CIDO argue, most of which are domain experts in different forms of epidemiology, viral vaccines, or we try to contact someone who is an expert and have them as consultants. And then that's one aspect where we try to decide. Second, I mentioned CIDO is relatively conservative with adding new data, which means that we try to require a fair amount, a great peer-reviewed scientific articles in order to add specific axioms or results that have already been accepted. So again, that's one of the boundaries we're going on for there. The second is that if we have cases of weaker associations in object properties, so stuff instead of causal relationships, we have associations or we have susceptibly causes, which we use as relations to show that there is some sort of fuzziness or correlation that we know exists, but we can't easily as always quantify as being axiomatic true if we say that it's a causal relationship. So for those, we try to find an appropriate relation to accurately describe it. Otherwise, it's mostly a matter of arguing with the domain experts that are involved in this project about whether to include it or not. And I'm going to mention a call to arms to anyone who's still working on this subject and wants to contribute and help. We welcome it. But I was going to save that for the end.
I mean, clearly the pandemic's over, right? I say sarcastic, so.
There's an argument that it is epidemic now, so instead of pandemic, so
Yeah,
still relevant, but
yeah, I'm being sarcastic, but yeah,
unfortunately, I'm not paid to be sarcastic,
yes, fair.
so. Okay, so I'm going to move faster since I have 15 minutes with all the questions. So one of the other examples that we want to bring up in via representation is with the Comirnaty vaccine or all the licensed viral vaccines. So CIDO imports all these terms to Project Violin, which is linked to our own vaccine ontology, which eLab runs and supports for what's going on. And each of these results as part of the COVID-19 vaccine database has a bunch of minimal knowledge standards about alternative names, manufacturer as specific instances, in addition to specific clinical trials, which we can link up with metadata results. So while we have all these results in ontology, we can also link up the ontology to our own knowledge base or database if we want to go into further details or handle some of the instance level results. Otherwise, the ontology gets really bloated and takes a long time to load. So while we have the database being linked together through the use of ontology IDs to map across each other, we can also take a look at that as part of our ontology tool and then just access the specific VOID if we want to query more information through any of the existing knowledge base.
Yeah.
Yeah. I don't want to go over this, so I'm going to speed up a little. So when dealing with clinical metadata, we have specific terms that we use to represent individual people, specimens, properties that are assigned to any clinical trial data sets. Even if we don't have the specific instances stored in the ontology, we still have a systematic framework to represent all of the metadata. So if someone wants to do a query or try to represent these in CIDO, we can have a consistent schema across all of our query results. Okay, just keeps moving. The other one that I wanted to focus now is that we focused on the actual representation of amino acid variants and some of disease epidemiology. But we are now more interested on the fundamental interactions of the COVID-19 disease process. So we're focusing more on these host coronavirus interactions. Now, I'm sure everyone is well aware that the S protein is one of the more significant proteins, both in having its interactions with human ACE2 as part of mediating the mechanisms for viral entry to the cell, in addition to interactions with angiotensin 2, which are linked to some of the damage that you see in capillaries and blood vessels. So, in addition to having specific axioms describing these results, it also has multiple parents, reflecting some specific other axioms and interactions that we get through our terms. I also want to mention that when focusing on this protein, we can use we can shortcut axioms to link up interactions with other proteins. So there was a paper published back in 2021 about analyzing the effect of cross-reactivity in immune epitopes across various coronavirus species. So as part of the hypothesis that was the initial explanation, some of the variation of why people do fine or don't develop vaccine. And so to show that there's some relationship such that we have, if exposed to a common cold, you have some small immune response or protection for SARS-CoV-2. Still smaller, please get vaccinated if you haven't already. And we can still show that relationship through mentioning as epitope cross-reactivity. And going from this host pathogen interactions, we can also move into protein-protein interactions we get between the actual human host and viruses, which we can use to take a look for some of the actual interactions for binding processes that we have experimentally observed. So in this case, if we have a specific event of binding between NSP5 and human HDAC2, we know that whenever those proteins were to interact, then we would have a specific participants for being involved. And then within this process, we know that they would inhibit each other as far as their binding. So while we don't have specific terms relating to the frequency, we can still cover these PPI stuff as part of ontology. So we have a bit more stuff where we do some handling of different gene signatures if we want to instead look at gene enrichment instead of just protein-protein interactions, where we link up specific genes to qualities of phenotypes and describe susceptibility to specific diseases or regulation. This is what GEO tends to be mostly focused on, or some of the other related ontologies as part of biomedical investigation ontology, I believe. So we have that framework for both genes, and then a bit more complex one when it comes to protein. And also, if we want to handle other aspects of the host phenotype, such as sex differences, we can also include that as part of qualities for specific patients. Okay, skipping ahead. So when it comes to the visualization of CIDO, one of our contributors have this weighted aggregate taxonomy tool in order to try to make much more sense of CIDO and make it easier for people to understand where we focus on in terms of information content and the actual representations. So while it's a bit too clustered for what's going on, I can still handle some of these boxes. So each of these boxes represents a category of classes which have a set of common axioms or relationships that are together. This includes both based off of child and parents, but also other groupings. So you can see we have a cluster of diagnostic assays in the upper right. And on the left side, we have a series of infectious disease processes for what's going on. However, the bulk of most of our information when it comes to CIDO are relating to chemical entities. So this is relating to the viral proteins, to the amino acid variants we tag to the emerging for SARS-CoV-2, in addition to the host proteins that we added as part of CIDO. Of course, all these relationships have their component information from CIDO and also interaction networks, but that by far represents the biggest dance of CIDO. So,
What is the color code?
so each of these, the color boxes are arbitrary. So what's going on? So yes, that's what I remember. There was a preference for related terms to have same colors if they're children, but that wasn't necessarily. I think it was this. I forget the algorithm used, but it was algorithm make this distinct and pretty. But each color otherwise represents a specific category of related terms that share the same ideas or axioms together. So the specific colors themselves are arbitrary, but each box represents coherent entity. If you take a look at the numbers, those represent the breakdowns for each of the quantity of those that are either direct children terms, any descendants, those that are bottommost children. So since this is okay, I have 7 minutes and I have 4 use cases, so I'm going to try and speed this up. So we mostly want to present 4 major use cases when it comes to CIDO. The first one, which I think I've exhaustively shown, is how the ontology-based coronavirus-related knowledge and data standardization is applied to a wide variety of different fields of CIDO. So I don't feel the need to belabor this point much further. The second, I want to show off the use of the query feature for ontologies, which we can take a look at some of the inferencer to try to identify potential amino acids, proteins, or other features that we would be interested in investigating. Third, I want to show an example of using CIDO to aid natural language processing to understand basic clinical and mechanistic research. And finally, we're also using CIDO's annotations in order to try to guide some machine learning predictive drug cocktail design for COVID-19 treatment. So one of the web tools we use with ontology is Protege, and it contains a very useful DL query, which if we type in on top over here, we will the specific relationship. It will go through the ontology and then look through any inferences if any equivalent terms. So in this case, we're looking for any mutations or variations of SARS-CoV-2, Omicron, and Delta that are shared between each of these species according to CIDO. And of course, you can see that we have 4, while if we take a look for either Omicron and Delta, we have much larger amounts of 45 and 18, but in CIDO we have 4 overlap. I will point out that the reason it says 4 or 5 is because there's a 5th result or of nothing, which always pops up, but that's not relevant. So taking a look at these specific SARS-CoV-2 proteins and looking at them in the literature, we find that almost all of these tend to have very important functions with either virulence, transmission, or greater affinity to the actual SARS-CoV-2 protein binding. So 2 of these, ST614G and ST478K, both mostly involve viral mediated entry into the cell, while the polyprotein amino acid variants PP1AK56R and PP1BT478K are those linked with viral replication. So while we did that as part of the query, one of our further results were, instead of just focusing on actual specific viral variants, we would instead take a look at some of the specific amino acid domains for SARS-CoV-2 and see if there's specific proteins that are within the same functional domain or same range for the actual protein domain and have shared with specific viral functions while mapping using geo terms. So while we can see that each of the S1 and ST cell epitope domains are fairly diverse, we see that there's roles for viral spread in parts of the immune evasion, also with some reduction in vaccine response. We can also see specific amino acid variants and other proteins playing different roles in immune evasion. So That includes with monoclonal antibody immunization as part of the SARS-CoV-2 PP1A protein, but that corresponding with the protein names. But we also have ORF9B, which is one of the putative proteins that we had previously identified. Actually had a set of deletions which were shown to suppress some viral autophagy, which was one of the arguments I remember saying that maybe we should update this into full protein through as something that's actually being described. Now, when we're using CIDO as part of the NLP program for SciMiner, which was used by one of our contributors, we end up getting this very interesting gene-gene interaction network. So each of the red diamonds represents SARS-CoV-2 genes. The green proteins represent specific genes that we have as part of the specific literature of CIDO have been experimentally shown to show up. Specifically, it was any part of the NLP said that this gene interacts with this later gene. With the blue genes popping up are those that are not directly linked semantically as part of the NLP, are still linked into other studies with SARS-CoV-2 papers to each of the human genes.
Yeah.
So you can mostly see that with the viral immune response, which is blue, where we get to have a length of the blood of additional interleukins as part of the host response against the virus.
This is real tough because, it's cell specific.
Yeah.
Like that. It's, modeling host pathogen interactions.
Yeah, and of course, I also want to bring up that one of the ideas for part of the damage that SARS-CoV-2 does is due to the cytokine storm being triggered by
Yes.
interleukin response. So part of that which is being captured here with the pathways that have been shown to enriched. Yeah, I'm trying to hurry up, so can go back any questions. Fourth use case was relating to the use of SARS-CoV-2 drug screening on a drug cocktail strategy. So we collaborated with another lab which converted CIDO into a series of vectors, and then used that to infer specific relationships for potential proteins based on the actual functional annotation. So the drug cocktail strategy basically states if we combine a bunch of drugs with similar effects, that might end up being more effective than if they were by itself. And while we get a bunch of shared interactions, we did, the paper did identify two specific chemical compounds as part of CIDO, which based off the screening should be able to interfere with 4 different roles of SARS-CoV-2 and will be a good potential drug target. So Octinomol is, I believe, a derivative of an anti-malarial, which was part of some earlier research and focus on potential protection against SARS-CoV-2. Now, so in conclusion, CIDO integrates a wide variety of terms for coronaviruses and terms related to coronaviruses. CIDO supports computational analysis of coronavirus and disease data, which can be seen on both the drug repurposing applications, and also for potential identification with epitopes and vaccine efficacy. And then we have represent some of the more important coronavirus mechanisms that are crucial for our progress for further research and work against COVID-19. Again, I want to reiterate that any collaborations are welcome, especially if there's something that you think is missing or could be improved on the CIDO. You can find us on GitHub. And while I don't think I'm pressed on it, this was a monumental task where we had many members of the community, both as consultants and as LBO researchers. So including Brian over here, so thank you. But these include faculty, students, and a wide variety of different positions in the ontology community and healthcare fields. Here's our funding for what's going on. And this talk was basically switching down to 4 different conference papers and 4 journal papers that we've worked on through the last 3 years. Whew. Yes, sorry, I went a minute over. Are there any questions?
Thank you.
Okay.
Remarkable amount of work.
Yeah,
So I have a question. So you indicated in one of your slides that
it's version control, how do you establish what's enough of a change to update the version?
So there's the old method, which we did initially, and then there's an official release method, which we're supposed to be followed. So internally, we typically increment version control for one of the minor versions whenever someone pushes an update to GitHub, which is usually done after discussion with some of the committee members and then substantial changes. Substantial being not particularly defined, but more than like one individual mistake. We have an official versioning control that we're supposed to follow along with other OBO boundaries, which we're supposed to do for significant major updates, but I don't think we've quantified what constitutes a significant major update. So right now, it's also that when it comes to a lot of these other ontologies, normally we expect smaller updates of, hey, can you add this or do that, which we add internally as increment up for our version control. But otherwise, we don't have we're supposed to have a strict criteria, but we haven't laid out the specific details of the strict criteria for major version updates.
And I also wanted to ask, how is this made available to users? I know that you mentioned that there's a query function, but is it also available like a download? And if it is a download, how big is it? Because
Okay. So to repeat Eric's question, and I should have done that earlier, he was asking how is CIDO available for when it comes to using tools and having accessibility. So CIDO itself is available on GitHub. So anyone that they want to take a look can download. As for the file size, there's two versions of CIDO. We have the base CIDO edits for what's going on, which I think it's like 8 kilobytes. I need to double check because I honestly do not remember the specifics. Which we have a base one to edit, and then we have a series of other terms that we import from other ontologies. And these are the related axioms, which are not necessarily needed as part of the CIDO specific axioms or references. So with imports, I think it increases by like 5 or 6 times as much. So I will say that currently the launcher version of CIDO requires the use of ELK or other efficient reasoners if you want to, or specific tools if you want to reason and query through it in effective time. As for looking for the ontology proper, we use Protege, which is this ontology web development tool, which you can make things easy to add specific terms, or also to use a reasoner or make these DL queries. I know you can also structure specific Sparkle commands or Sparkle queries if you want a more succinct approach, if you're just given the raw OWL file. So that is an option. Lab also supports the use of Ontology, which is a web tool which allows you to just query specific terms and you just get to see the hierarchy without downloading. So we try to provide a bunch of tools to make it accessible and for people to use.
And then last but self-serving question, what was your criterion for drawing interactions between host-pathogen interactions? Specifically PPIs.
I believe that they are from specific primary research articles that have shown experimentally concluding the results. If you're asking what were the criteria of that specific papers, I don't know offhand because I wasn't involved with that specifically. But it's primary peer-reviewed articles. I know that we talked early on about potentially including preprints, but I argued no. So it does mean we're kind of slow with emerging threats or other details, but we've tried to adopt strategies to handle it.
The reason why I ask is because there's a very strong difference between something that is observed like a crystal structure showing the interaction versus something that just showed up in like a pull down.
Yeah.
So
again, it's a case where I would have to take a look at reference that was used since we have it, but I don't know offhand.
I think we have a question online from Matt. You want to go ahead and ask?
Yeah, hi. Really an impressive amount of work, really broad. It seems like a super ambitious project to organize everything around coronaviruses with the you said thousands of papers have been published over the past couple of years here. I'm curious on sort of how you came about your the scope of the project, and how do you know if you've like bitten off too big of a piece to chew? Like how would you know that you're being successful? Like you could picture that just your ragtag team is not going to be able to do everything. So like, you know, how did you decide to go so big rather than something more manageable, like say just focusing on a part of the sort of complex landscape around coronavirus infections?
So for Ambition, ask Oliver, since he was the one who decided the initial scope of it. But no, I think part of it was that this is a big field, that's something that's needed to be grabbed and handled, but there was a problem that needed to be addressed in order to deal with all these issues. As for getting issues with it being too big to handle, currently, from my work on that, I know that I've gotten much more narrow with my focus since early on. I tend to help with more of the high level structures. While for the past year or so, I've been much more narrowly focused on the amino acid variant side story. So I know that in these cases, I've definitely narrowed my scope to something that's a bit more manageable, simply to try to represent something significant. But as for the others, well, we ask for contributors and help to both because we know there's more to be addressed and that we could use other people's aid. So.
Yeah, for sure. I can add some. Yeah, definitely it's very ambitious project. Meanwhile, it's also community effort. Right? So you are actually it's not only one group. It's more like a bigger community. So, for example, the protein ontology work. I mean, basically, we didn't do it. We just asked our protein ontology group, right? Like NATO. Yeah. So they provided the information for us, and we have the mechanism to systematically, easily, like seamlessly integrate into our system. So this is how you design and reuse. So and then it's not only we, it's like more like you come into us, and then we come into the whole world, right? So we put yours in, and then this one is also openly available for the whole world. So yeah, definitely it's ambitious. But we're trying to, there are actually a lot, probably 7, 8, or even more than 10 ontologies, somehow working on different levels of COVID. Yeah. Definitely, I think ours is probably the most influential one. And a couple of others actually have merged their terms or their domains to our ontology. It's becoming more and more a community effort than before.
Yeah, I don't know how much you heard that, Matt, since Alvaro was sitting in the back. But the answer is that we have a lot of help from a lot of gifted and driven people. Oftentimes, they are working on a specific subniche or stuff with coronavirus. We talk to them, and then assuming that everything aligns, we help integrate it into CIDO. But it's truly a matter of get more people, get the community involved to help with the new terms, and then we just add them as we come.
So given the time, I think we probably need to take any additional questions offline.
Yeah. Thank you, everyone, for staying on past the end. And everything looking pretty good.